Okay, so we have talked a lot about the key driver analysis. So here actually I just give a little bit more details how we are going to identify drivers. So the idea actually started from the paper we published in 2008 in Nature Genetics. In that paper actually, Zhang formally integrates multiple types of information like genetic, and the gene splicing, and the transcription factor, and the protein interaction. So almost all information actually available to us. And so we found out that integration of all those types of data actually can give us the most predictive networks. And so this actually, the key driver analysis shown here is actually a more formal presentation of the method. So okay, let's start from simple. So basically, you have a network like this, and so you can define links directed or undirected. A link can be weighted or unweighted, and then you can define like collectivity or a bunch of centrality and closeness and a degree and a page rank, and then you have module. The modules can be overlapped or can be separated. So those are the basic concepts. So the goal is try to identify those hub or key nodes in the network. So in those social networks, actually, it is pretty looks pretty simple. So you can easily define those the driver genes like the hub nodes. But in the biological networks, actually, is more complex because a lot of links actually inferred based on our network approaches may or may not be true because we have limited information about the biological system. Imagine that now most of the data available is static, so it's kind of a snapshot from each sample at a particular time point. So, but the real biological system actually keep changing all the time. So every second, every millisecond, so you see this kind of changes. So we have no means actually fully recover those information. So it means then this actually means the networks we constructed are noisy. So to compensate this kind of noisy networks, we try to put together multiple types of networks. So we call like a multi-scale approach to identify like and the true signals and also the key regulators for those subnetworks of our interest. So this is a figure I showed actually earlier. So we have the co-expansion networks. So you derive modules, either overlapping modules or non-overlapping modules. Then you have a causal network. And when you project those modules onto the basic network, and then through the key driver analysis, then you can derive actually simplified subnetwork. Okay. So this is for most presentations I shown previously. Actually, was based on this simple schema. So you just integrate the modules from the WGCNA and the basic network, and also sometimes the causality network based on pairwise causality test. So we see a lot of applications, and this is the core procedure for the key driver analysis. So you have an input gene list, either from like a module from the WGCNA analysis, or from the differential gene splicing analysis for case control. Samples, and then you have the causal networks, and based on another large scale genetic or genomic studies, and so you want to understand which genes in the gene set or any other genes outside the genes, the gene list, are the drivers for this particular gene set. So the answer lies in the network we are going to use. So it is important actually to have relevant networks here. You basically, so protein interaction network probably is the most frequently used network for almost all studies for different diseases, but that is probably not tissue specific. It is not disease specific. So even though you can get some signal from there, and also the same, but it's probably is still quite general. So the same actually holds for the ingenuity and many other public databases. So they are not disease, not tissue specific. So in this case, actually, we have to be very careful how to make use, how to select and networks. So there are many actually, many packages actually available for building up those causal networks. Like in R, I know there's a package called like DEAR. You can use it for building up a basic network, and Alias has also the causality test packages. So you can use all those approaches to build up networks based on the data relevant to your study. So once you define the gene site and the relevant causal network, then the first step is to define the subnetwork like around this gene site. So this basically we try to restrict the answer to a smaller network by looking the neighborhood of the around those input gene site. So now we have two types of approaches for key driver analysis. One is the dynamic search. So basically, if the input nodes actually is only a subset of the network, like NG here, it means actually those genes actually they could some of the genes actually could link to each other through many other genes outside the gene input gene list. So in this case, we have to figure out whether actually those genes actually of our interest is really enriched for in certain actually neighborhood. So I will give more details about this. So basically, you want to check whether actually some nodes, neighborhoods are more enriched for the gene list. Then if there is a significant enrichment of the downstream neighborhoods of a particular node for the gene site of our interest, then we claim actually this particular gene will be a potential driver for the input gene list. But on the other hand, if the input gene actually we don't have any input gene list, and so you just have the network. So what can we do? So then you basically just look at the downstreams and also the immediate neighborhood, like degree or R degree, to rank order the genes in the network, and then identify the hub genes as the global or local drivers. So again, I have given more details.
Ben,
yeah.
A question. Could you go back to the slide and explain to us these acronyms? What does NGH stand for? Do you see where you say static MGH search?
Yeah, so NG actually first, it is a,
No, it's on the side. It's a dynamic NGA search and a static NGA.
yeah, NG, yeah, this kind of is a neighborhood. It's kind of, it's a neighborhood search, yeah.
I see. And
Yeah.
what does NPH stand for? Do you notice the right? I tried it by NPH.
Yeah, it should be a typo here.
Oh,
Yeah,
okay.
yeah.
NGA.
Yeah, it's just a abbreviation of a neighborhood. Yeah,
I see.
yeah. It's,
I see.
yeah. So for the dynamic neighborhood search, I call like a DNAs. So basically, so you have an input gene list, like we call G, and a network called N. So the first step is to generate a subnetwork, NG. So so you can expand the network actually surrounding the input gene list. So because sometimes actually the causal network is quite complex, so we want to simplify the problem. So we limit the search within the neighborhoods surrounding the input gene list. And then we search actually the optimal layer, the optimal layer whose neighborhood or is most enriched for the input gene list. So okay, so you start with like a layer 1, so you can check the enrichment. So in this case, for this yellow nose, so you see actually it has 4 or 5 actually immediate downstream, and 3 of them actually are in the input gene list. So you can do the enrichment test. So probably is pretty high, and the enrichment p-value should be very significant. And then you search the 2 steps neighborhood, so you can see actually more the genes in the 2-step neighborhood. But when you calculate the enrichment, probably the p-value will not as significant as the first layer neighborhood. So we could just keep getting all the statistics from every layer neighborhood, and then find out that the best layer actually whose neighborhood most enriched for the input gene list. I hope this is clear. So it's a very simple concept. So you just dynamically search all possible neighborhood and figure out which neighborhood is the most informative. And then we claim if yeah.
I don't understand how the sub-network grow up. It's grow up from a point and just expand or just...
Yeah, you start with every node in your input gene list, okay? Expand like for certain layers, like you can so you can make choice like 6 or 5, whatever, 2 or 3. So you expand. So for every node you expand. Yeah.
When you design your algorithm, the node selection, is there a random selection or just everyone?
Node selection, yes. So you're talking about the input nodes. The input nodes actually should be biologically meaningful, either from like those modules derived from WGCNA, like the inflammatory gene signature. You can derive from the modules enriched for immune response genes, and is also actually most relevant to obesity diabetes traits, or you can derive from the differential expression analysis. Those genes actually are informative or associated with certain disease and status, disease state. So it's not random. But when you expand the network, actually, you treat every node equally. So because we don't know which nodes actually are more important or not. So just look at the neighborhood of all nodes, and then you have a expanded subnetwork surrounding the input gene list. Okay, so now once you found the optimal neighborhood, then you can, okay.
How do you determine age?
Yeah, that's a very, very. You can, so in the package I will show you in the afternoon. So I have some default value, but you can make any choice you like. But this again, like, is something we need to optimize. But so far, actually, we don't have a good idea how to do that because, we don't have a good validation data set to cover so many drivers. So like for the disease systems, we actually just saw in the previous presentation. So for every network, we have only 1 or 2, at most 10 genes actually locked down, and so in no way actually we can have a gold standard to test. Yeah, but this is something actually we should do, and so we hope that for this community we could work together to get some gold standard and data to validate those networks. Yeah.
What enrichment statistic do you most commonly use?
Say again?
Which enrichment statistic?
Yeah, you can use like a feature exact test. Yeah, that's the commonly used. Yeah.
And then you just use the same age for every, for the entire network, or you don't define a new age for each?
You mean maximum? Yeah, that you can search it from. Yeah, you can use the maximum distance in the network, or you can restrict to certain maximum layers. Yeah. So all actually those are the parameters you have the freedom to set up. Yeah. But again, like it is a problem of optimization. So we need some gold standard to figure out. But so far we don't have much data about this. Yeah. Yeah. So basically, then we claim those. Yeah.
Sorry, quick question about age still. Do you find that in general the key drivers are robust to choice of age? So if you find a key driver with your best choice of age, it still looks like a key driver for other age?
Yeah, normally actually it is like that. And also we have the data published actually shown that actually those immediate neighborhood tend to be most important.
Okay.
Yeah. Yeah, so then actually you can derive those candidate drivers whose neighborhood actually is significantly enriched for the input gene list. And then from those actually candidate drivers, actually we can further differentiate those global drivers, like those drivers without any parent nodes. So then they tend to regulate many other nodes, but and without any without being regulated by other nodes. And so the rest of the nodes actually we can claim as local. So again, like this kind of definition is not fixed, and it can be actually modified, and if you have some gold standard to test. But all those actually is really actually based on empiric experience. So this is the dynamic. because you have the nodes, the input nodes is only a subset of the nodes in the network. But on the other hand, like if you don't know the relevant gene site, you just have a network, or you have your gene site actually is a is all actually basically your gene site actually is a list of nodes in the whole network, and then we can use a simpler search. So basically, you just look at the downstream and also look at the immediate neighborhood, and you use the kind of mean plus standard deviation or two standard deviation to define those the global drivers. Yeah. Okay, so we have a few applications. The first one is application to the yeast system, and because this system has been actually comprehensively studied, and we have built up the basic network in 2008. So in this paper, actually, we basically search all the downstream and overlay those downstream with the EQDR hotspot. So basically, the EQDR hotspot basically is those loss actually as a collection of loss actually associated with many gene expressions. So basically, in tradition, actually 13 chromosome regions actually were identified to associated with a large number of gene expression studies. So we call like EQDR hotspot. And so we also build our basic network, and through integration of many types of information. So in this application, so we use this key driver analysis, especially the dynamic neighborhood search, to identify the drivers of those EQDR hotspots. So the input for this key driver analysis actually is the list of genes underlying each of the 13 EQDR hotspot. So this is the EQDR hotspot, and that's the y-axis shows the number of gene expression associated with a particular locus. So you can see those hotspots along the whole genome. So this is the basic network. So it's the yeast system is relatively simple. So you can see the basic network is relatively sparse. But in the human study or mouse study, you can see a much denser basic network. Then we actually use the dynamic neighborhood search to identify those drivers for each EQDR hotspot. And so those are the drivers that we identified. So I give you more details in the next few slides. Okay, for like EQDR hotspot 11, so in the original approach, so we identify only SOR1 as a key driver, but through the KDA procedure, we identify 3 additional drivers, and also we see like like ISM25 new driver actually is more significantly enriched for the genes underlying this EQDR hotspot. So basically, actually, it means potentially ISM25 is a more likely another global driver for the whole system, which actually missed by the previous procedure. So there is another actually hotspot trial actually, yeah. Again, like a PHM7 is the only regulator identified by the original approach. So here actually we identify 6 additional regulators, and actually 4 out of the 6 and the new drivers are much more significantly enriched for the genes linked to the hotspot than the PHM7. Yeah, so basically these 2 examples just show the power of the KDA approach. Then the original and the simple downstream overlap with the EQTL and the hotspot. Yeah. Then we come to the inflammatory driver genes. So in this particular application, so because the gene list is quite long, so we have like 2,500 inflammatory gene signature, and also the basin network is quite dense because we have we just simply using all the tissue-specific basin networks. So from like 10 to 20 actually large-scale genomic studies of obesity diabetes. So yeah, because of this actually, in this case, so it makes more sense to use the static neighborhood search. So basically, you just look at the first step, you limit the network to the direct links and between any 2 genes in the gene list. In the signature, we identify through this the 12 and the mouse model. And then you derive those inflammatory signature specific and also tissue specific network. So you can see here. So this actually becomes much more sparser, and then we identify a number of conserved the drivers as I shown before. And so again, like we validate those drivers through the MGI database. So the top drivers and the global drivers, and they have the importance in descendant order. So that's what we found out from this kind of key driver analysis of inflammatory gene signature and the consortium of a basin network. Yeah.
Can you explain that in the application to when your inflammation driver gene, those inflammation driver gene what is derived from all the I think study was in the mouse, right? Mouse or rat model, and then you were using those in human data, right?
Yeah. So the example shown here actually, yeah. So the signatures. The inflammatory signature was derived based on the mouse models, 12 mouse models here. And so the 2 examples actually shown here is human. Yeah. There's a liver and adipose from multiple large-scale studies and gene splicing studies.
So are you using those networks that was discovered in human mouse, that you are using those genes in human data to find out their property?
Yeah. But because actually they all actually, both human and mouse data actually were used to model the obesity diabetes, and because we also found that in this human systems, those inflammation is a top driver of obesity diabetes phase. So this and 2 studies, 2 types of data are quite relevant in this sense. Yeah, that's what. Yeah.
Do you think that the genes which are involved in inflammation in those mouse models will be about same in
It's not the same because they're conserved in somehow. Yeah, they definitely you may see like a 30 or 40% conservation, but there are still a large portion of genes actually specific to mouse system or to human system. So that's a good question because now we look at the commonality between mouse and human, but then there are some specificity and for different species, but we didn't look at those. There could be actually mouse specific drivers and human specific drivers. Yeah.
That's a continuation of previous question. Is there any kind of the model consensus or preservation of key driver statistics for this analysis? I mean, just do you have any kind of statistics to see whether this model or your identified key drivers is preserved in another analysis?
Yes, actually, yeah. Now you can see actually from here, the adipose and the liver, those are both from human, and like a thyroid BPH, CK, CD55, that are drivers. We see actually the same set of driver, actually many of them also in the mouse networks, yeah, and across species and across gender, yeah. But then they are again like those are the common drivers. But for they are also like gender or tissue specific drivers. Yeah.
Yeah. I know.
Maybe I can also add something. I think what you may have in mind is something like a module preservation. The thing is, remember our module preservation statistics in WGCNA are really for undirected networks.
Yeah.
But I think it would be a wonderful research endeavor to generalize them to directed networks. Yes. So if somebody's looking for a dissertation project or it's a good paper.
Yeah, it is. Yeah. So definitely there are a lot of open questions. Actually, we don't have time and don't have energy to explore because the network is just so complex. When you publish like a cohort, we spend like millions of dollars. So you just explore a little bit of the biology. So actually, I would say like 99.9% of information actually completely wasted and without any further understanding. So that's my understanding. So like for all the data shown here, actually at Rosetta, I think we spent like $100 million, but eventually you see just a few like driver genes. So just kind of a waste. Yeah.
But the good news is the data are available at the SAGE website.
Yes, SAGE. That's actually the motivation for setting up at the institute, so that people around the world can make use of the data, make use of the model, and for validation. Yeah.
Ben, before you move on to the cancer, can you say something about this inflammatory gene signature that you've identified in liver and adipose and various human cancer and muscle? Is it a reflection of macrophages that have infiltrated those various tissues? So what you're really doing is tracking macrophage gene signatures that are deposited in the various tissues that you profile. Can you speculate on that? Because all of these tissues are heterogeneous, and we deal with the same thing when we profile islets. It's many different cell types in there. And if something changes, is it really because you've introduced a macrophage that has landed and grown in that tissue, and that's why you're seeing this repetitive signature show up?
Yeah,
that's something about that.
Yeah, that's right. Also, actually, is the limitation because most of the time we thought those immune response is a kind of passive and downstream. So for the upstream, especially for the Alzheimer's disease, probably there must be some actually upstream event trigger this kind of immune response. But so in this case, we should pay actually attention to the subnetworks actually down the list. Like in the Alzheimer disease example, I will talk a little bit more in the afternoon. So I believe that even though actually inflammation actually is the top ranked the module for AD pathology, but I believe actually those neuron related or cell cycle related those modules actually specific to neuron or oligodendrocyte, they are probably more likely to be the upstream event triggering the immune response.
Maybe we resume the discussion. I think you made a very good point.
Yeah.
Also then maybe just proceed.
Yeah, so we have just a few minutes, so I can quickly wrap up. So for the cancer network, so we actually, because these regions actually includes a small number of genes, and so in this case actually we use this dynamic neighborhood search, and then we can get a sparser and a Bayesian network. So again, like many of the genes have been validated through the siRNA lockdown experiments, but even larger actually portion of the drivers have not been validated yet. And so here actually in the middle is alpha-1. So we have a paper actually in preparation to show that this particular gene actually really impact proliferation and migration. And also we shown that the predicted subnetwork controlled by F1 actually is highly enriched for the experimental signature of F1 lockdown, siRNA lockdown. Yeah, so again, like we shown those drivers are more important than the non-drivers. So in the Alzheimer disease, so basically we found a similar trend. So far actually only 13 unknown AD susceptibility genes actually were identified through the GWAS study. So we believe those are the upstream genetic and perturbation for the disease. And so in this case, actually, we see those AD GWAS-derived genes actually enriched in the drivers that we inferred. So basically, those drivers are actually 9 times more likely to be the non-AD susceptibility genes than those non-driver genes. So this is just the one step to advance our selection of a potential driver genes for Alzheimer's disease. So again, like for TyrBP, so we have the perturbation TyrBP, and we shown the huge enrichment of the TyrBP experimental signature in the predicted network. Okay, so in summary, so we this simple actually KDA method actually was developed to identify the key hubs of a biological network, and based on the downstream and the degree centrality. And so the drivers actually in multiple disease systems have been shown to be more important than non-drivers. And many actually drivers key regulators have been tested or validated in the different ways. So we also have a large scale validation system to show the relevance of the drivers. And but again, like this is a simple method, and a lot of other actually centrality, like betweenness or closeness actually can be used to explore the biological importance of the drivers, and that is actually the last step. And the two people from the Sage, by another words, I contribute to the package development and the Justin and Dave. And again, just as the key driver analysis, it's very simple. Yeah.